So, good morning, everyone. We are the group one, and for today, we're going to do the pre-lab of this laboratory activity number 8, which is all about the isolation of carbohydrates, yung liver glycogen, and plant starch. So, next slide. So, for introduction, carbohydrates are called simple or complex depending on their chemical structure. So, na lesson naman natin to sa biochem lec na ang building blocks ng carbohydrates yung mga monosaccharide, disaccharides, and polysaccharides. So, meron tayong mga simple sugar, which is yung monosaccharide, and complex, which are polysaccharides. So, di ba, parang continuous na monosaccharide, kaya parang complex ang kanilang chemical structure. So, simple carbohydrates include sugars found naturally in foods such as fruits, vegetables, milk, and milk products. So, we have those yung sucrose, fructose, lactose. So, yun. So they also include sugars added during food processing and refining. So complex carbohydrates, which is yung mga polysaccharides natin, include whole grain breads and cereals, yung mga starchy natin na vegetables and legumes. So many of the complex carbohydrates are good sources daw of fiber. So next slide. So for this laboratory activity, we have 3 objectives na dapat ma-accomplish natin by the end of the laboratory, which is i-conduct natin next week. So first, we have to prepare the potato starch. Second, we have to isolate the glycogen from the chicken liver. And last, we have to test the presence of starch and glycogen. Next slide. So for the materials, ito yung mga gagamitin natin na materials sa lab. We have the grater, knife, the cheesecloth, beaker, burner, filter paper, test tube, medicine dropper, steering rod, wire gauze, iron stand or iron ring. We have the chicken liver, potato, distilled water, iodine solution, 10% glacial acetic acid, yung mga Molish reagent and Benedict's reagent, 95% alcohol, and filter paper.
And now let's proceed to the schematic diagram for each procedure in this laboratory activity. So for the first procedure, which is the preparation of starch, the first thing we need to do is to peel the potato after washing it and choose either to grind or grate it. Afterwards, mix it with 100 ml of distilled water in a beaker and stir it thoroughly. Next is to strain the pulp using a clean cheesecloth while catching the liquid in a beaker, and allow the starch to settle and discard the supernatant liquid. And lastly, wash the starch using the decantation method, and lastly, strain it and set aside to dry. For the second procedure, which is the testing of starch from the previous procedure, the first thing we need to do is to place 1 gram of prepared starch from the previous procedure in a test tube along with 10 ml of water. Next is to form a suspension by shaking the test tube vigorously and pour the suspension into 50 ml of boiling water in a beaker and constantly stir the solution and allow it to cool afterwards. Get 2 ml of sample from the cool starch solution and pour it into a test tube and add 3 drops of our iodine solution. And lastly is to record our observations in terms of the appearance, color, and maybe odor of the solution.
So for the next test is the isolation of glycogen from chicken liver. The first step is to wash the liver and pat dry with filter paper and weigh out 2 grams. So next is you need to transfer the liver on a watch glass. And the next step is pour 6 ml of 10% glacial acetic acid in a mortar, then add the liver. So as you can see sa schematic diagram namin is dili siya ang actual na glacial acetic acid, so kay wala man na wala siya na sa lab, so nag-improvise lang may gikan sa sa canvas. So for example lang na siya, so mag-pour ka og 6 ml of 10% glacial acetic acid. The next is i-add nimo ang liver. So the next step is pound for 10 minutes. So pound is like yung i-crush mo good yung ano yung gi-add mo na 6 ml and then yung liver. So afterwards is add 10 ml of distilled water to homogenate, then let it stand for filtration. So next slide please. So for the last test, it is the test for presence of polysaccharide glycogen. So the first step is label 2 test tubes with control, which is the distilled water, and then the next test tube is for the liver. The next step is perform Mollish test, iodine test, and then the Benedict's test. So the last step is you need to observe the color of solution and record the results. So every test is need nimo siya i-observe if nag-change ang color or may odor ba or may precipitate ba na nag-appear during the test.
Wala ba si Rowelyn? Sige, ako na lang. So for the first guide question, why do you filter the acidified extract while it is hot? So filtering the acidified extract while hot helps to remove impurities and solids that may have precipitated out of the solution due to the acidification. And when you cool it down, acidified extract tends to precipitate, and then you'd be filtering off product 2.
Ito yung kay Lalane dito, no? So, what is the principal structure difference between amylose and kay Joem ata to.
Sige. For question number 2, what is the principal structural difference between amylose and amylopectin? So amylose is a straight chain polymer in which 1 to 4 glycosidic bond links with the glucose unit, while the amylopectin is a branch chain polymer in which 1, 4 and 1, 6 glycosidic bond links to the glucose unit. So according to BIOCHEM states that amylose and amylopectin are two different types of polysaccharides found in starch granules. They have similarities and differences, both structures and chemistry. Next slide, please.
Mo na jud ni final na jud ni. So, which between glycogen and amylopectin is more highly branched and broader structure? So, na-lesson din natin to sa BIOCHEM lec. So, under the sa starch, may dalaw tayong polymeric unit of glucose, which is yung amylose and amylopectin. So, amylopectin and glycogen, actually same lang sila ng structure kasi they both contain yung alpha-1,4 and alpha-1,6 glycosidic bond. But their difference is mas madaming alpha-1,6 glycosidic bond ang glycogen compared sa amylopectin. Sa amylopectin, mas madaming alpha-1,4 glycosidic bond. And because of that, kaya more branched ang glycogen kasi mas madami siyang glycosidic bond. So yun nga, as compared to amylopectin, glycogen is highly branched because it is supported by two linkages such as alpha-1,4 glycosidic bond and alpha-1,6 glycosidic bond between glucose monomers. So compared to amylopectin, glycogen structure is highly branched kasi nga mas abundant ng kay alpha-1,6 glycosidic linkages between glucose chains. So ito yung structure ng glycogen natin. So more branched siya kasi actually feel ko mas dami pa to. Pero yan yung kayang mas branch siya. Tapos makita natin, ito yung alpha 1,6 na linkage. Makita nyo man sa picture, tsaka yung alpha 1,4 niya. So next slide. Compared sa amylopectin, ito. Ayan. Feel ko actually madami pa ata to if parang small area lang yung parang ipakita dito. Pero if natin siya like yung parang ilayo good, kunyari yung sila itong picture, mas branchy jud ang glycogen compared sa amylopectin. Naka-share confirmation lang mo siya diri. Sa mo to. Next slide. So here tawag niyan. So dito na part is yung show the isolation of polysaccharide through schematic diagram. So according sa na-research ko, ito. So first we have to prepare the materials, which is yung potato, yung peeler natin, yung bowl, tsaka yung grater. So after that, we peel the potato, and after that, i-grate natin siya. Tapos after nyan, since na-grate natin yung potato, ilagay natin yung potato sa liquid, mag-add din ng 100 ml, and then i-store natin siya for 24 hours. So once pagka next day, after natin siya mag-store for 24 hours, yung liquid na yon i-filter natin kasi syempre may potato pa man yun. I-filter natin yon and let it stand until white solid settle the bottom. So after natin ma-filter na yung liquid, kasi so yung liquid natin, so yun, ipasettle din natin muna siya until yung white settle or yung white solid settle, yung white solid mag-settle at the bottom. Tapos after nun, since pag mag-settle yung white solid sa baba, slowly i-decant na natin yung liquid, and kasi may mga precipitate yan actually sa baba yung puti. Tapos after ma-decant yung liquid, yung parang supernatant liquid, i-dry natin yung precipitate. Tapos yung precipitate, yun yung polysaccharide. That's all. Thank you.